I'm going to be talking about biophysical signaling and carcinogenesis and the implications that it has for therapeutics. But in order to set the stage for this, what I need to talk about is the dilemma of cancer treatment today. Despite careful study of tumor biology and initial positive responses to therapy, cancers appear to have a virtually limitless ability to respond to treatment by reprogramming cells and developing resistance to specific therapeutics. If you look at the epidemiology of cancer mortality, what you will read lately is that cancer mortality is decreasing by 1.6 to 1.7% a year. But if you look actually, we don't have a pointer, do we? Anyway, at heart disease, from 1950, heart disease has greatly decreased, cardiovascular disease, infectious diseases. But cancer is basically been flat. It went up a little in the 90s, and so what you're seeing, the 1.6 to 1.7% decrease, is actually since 1990. Certain cancers, cancer of the uterus, the liver, and the pancreas have actually increased in mortality, as has melanoma in men. So to address this problem, there's been a change in the general therapeutic approach. Until fairly recently, cancers were treated with chemotherapies that were toxic to both malignant and healthy cells. Malignant cells grow faster, therefore, hopefully it killed the cancer before it killed the host. But to reduce collateral destruction, pharmacologic treatment modalities have gradually transitioned to small molecule therapeutics and targeting specific pathways. So the underlying assumption of targeted approaches is that carcinogenesis can be reduced to primary signaling pathways or genes associated with particular cancer phenotypes. If you knock them out, you'll inhibit the cancer. So success with these therapies we've had some successes, and the biggest one is with chronic myelogenous leukemia, where the BCR-ABL enzyme is in the constant on position. Tyrosine kinase inhibitor, imatinib, has been used to turn it off. It's also used in the KIT gastrointestinal stromal pathway. However, these targeted therapies also do collateral damage, and in this case, they damage p53 tumor suppressor genes. So, the body's natural defenses against carcinogenesis are apoptosis, DNA damage repair. There's like 5 different mechanisms. If you split the double helix, it can mend, it can replace base pairs. There are immune mechanisms and tumor suppressor genes. So if you stop to think about it, evolution has been a process of robustness and adaptability. If we really got cancer every time there was a random mutation, we never would have evolved. So there's a lot of mechanisms, feedback mechanisms in place to keep that from happening. So the number of signaling networks and epigenetic pathways involved in tumor etiology progression make it an extremely complex process, and the greater the number of gene products required to produce a phenotype, the less the effect of varying any one of them is going to have. So if you look at this, I need a pointer because I don't know how to use this arrow. Okay, this is a scale-free network. This is the way our networks look. Now, when you hear this business about cancer comes from random mutations, it sounds like a random network. You hit this gene, it hits that one, it hits that one, it hits that. So it's kind of like the old-fashioned Christmas tree lights. That isn't how it works. Gene networks and protein networks look like this. They're scale-free. If you hit this, most of these nodes only have 1 or 2 connections. You're not going to knock out the system. This is more serious if you knock out the central nodes. So in a sense, I show this to my students because this is part of a single reaction network of a metabolic process in Escherichia coli, which is bacterium. Now, do you seriously think that knocking out one single part in that thing is going to give somebody cancer? It's just not logical. Biophysics is coming. So because of these data, the hypothesis that cancer is driven by genomic mutations is being called into question. Evidence suggests that it is unlikely that one or several genes can exert pleiotropic powers over so many others and lead to a complex array of cancer phenotypes. So, accumulating data point to the overriding importance of dysfunction in the cellular microenvironment. Systems that exist to maintain health and defend against disease transition into dysfunctional states that instead support the tumor, which is why it can adapt to these medications. So microenvironmental cues, not single cells, are responsible for the dysfunction. And the field of epigenetics has made it abundantly clear that microenvironmental cues can both elicit neoplastic transformation i.e. if you expose stroma to a carcinogen, it transforms, and reverse tumor cells with multiple genetic and epigenetic lesions to normal in vivo and in culture. And what that means is you can take a cancer cell, put it into a normal animal, and it will revert. It's not the cell, it's the microenvironment. So, precancerous epigenetic changes indicate systemic involvement. Carcinogenesis is characterized by genome-wide hypomethylation and promoter-specific hypermethylation of CpG islands that precede tumor manifestation. And obviously what this does is this can lead to the inactivation of tumor suppressor genes and the activation of oncogenes. So this genome-wide hypomethylation shows systemic involvement before you ever see a tumor. So in fact, epigenetic changes have a stronger association with tumor progression than do specific mutations. Now, on the one hand, although we constantly talk about genetic causes of cancer, we give lip service to all of these other things increasing risk: smoking, obesity, diet, infection, inflammation, hormones, genomic variation, aging. All of those things cause epigenetic changes which influence the tumor microenvironment. Okay, so this is a quote, as you can see, from Lancet in 1962, and it's still as relevant today as it was then. Cancer is no more a disease of cells than a traffic jam is a disease of cars. A lifetime of study of the internal combustion engine would not help anyone to understand our traffic problems. So cancer is basically a traffic problem. So what we're doing is we're all looking at very specific aspects of cancer. And this is like the blind man and the elephant. You've all heard the analogy. The blind man who feels the tail says, oh, elephants are like ropes, or they're like snakes. He's describing exactly what he feels. The other guy is examining the leg and says cancer is like a tree, feels like bark. Another one says cancer is like a spear. They're all accurately describing what they see, but they're not getting at the essence of the elephant, are they? Okay. So abnormal cells can only thrive in a dysfunctional environment. Complexity in organisms arises from a large number of nonlinear interacting systems involving molecules, extensive crosstalk between signaling pathways, multiple feedback circuitries, etc., making it extremely difficult to predict behavior on the basis of changes in linear pathways. But that's what we're doing with targeted therapies. So the additional characteristics of complexity are emergent properties. And that means that a system has characteristics that the individual components don't have. So the best example I could think of is if you take a thermodynamically closed system, you have water in a closed jar, and you heat it up, you increase the pressure, right? So what is that? Basically, it's the movement of molecules, but if you didn't understand the concept of temperature or pressure, you could never predict it by the movement of a single molecule. One, because single molecules affect each other. They can either slow each other down, increase each other. So this is what we mean by emergent properties. So the conundrum of cancer therapeutics is that the whole displays characteristics that are not present in its individual parts, and investigating these parts out of context, i.e. in vitro, will not give you an accurate understanding of the way they function in an intact organism, nor will the targeting of single pathways be sufficient for changing complex interactions. So, as an example, the role of focal adhesion proteins in 2D cell motility, and I'm talking about the usual experiments in a petri dish, are not predictive of the role in motility in more physiologically relevant 3D matrices. Cancer cells in 3D matrices have fewer focal adhesions and stress fibers and do not form wide lamellopedia and associated pseudopodal protrusions. So, cancers beget mutations versus mutations beget cancer. One of the things we know is that the more progressed you are in stage 4 cancer, the more genetic mutations you have. And it's never made sense to me that people start looking at stage 4 cancer and start, seeing which genes are mutated and then creating drugs for it. I mean, the genes have not caused it. The cancer's caused the genetic mutations. So, no cancer exhibits any trait which cannot be found in normal tissue as the expression of normal genomic activity. No cancer grows faster than an embryo, nor is any cancer cell more invasive than a macrophage, nor are cancer cell lines more immortal than our germ lines. The only distinction is that in the cancer, The expression or lack of expression of many traits may be inappropriate for the tissue in which it occurs. Okay, so the human body, being a dissipative system, often functions far from thermodynamic equilibrium. Its systems are nonlinear, involving multiple interactions and feedback mechanisms. Homeostasis doesn't exist. Everything in the body is in a state of constant flux, and stasis is death. So, dynamical systems are deterministic systems which automatically evolve towards a state of equilibrium called an attractor, and this is where I'm going to go with the biophysics in just a minute. Once a new attractor has been established, the system will continue its evolution within the constraints of the system unless there's enough perturbation to move it away. It's kind of like a sinkhole. Once it gets in there, it's hard to get out. So this model unites genetic and non-genetic causes of cancer and explains the tumor's ability to adapt and circumvent interventions because it's an attractor. Okay, so cancer attractors can operate at a cellular or tissue level. There are many systems. A large number of elements in a system leads to complex behavior and multi-stability, stability of certain patterns of expression attractors. Mild perturbation doesn't disturb this pattern, and this is what I was talking about, robustness and adaptability. If you just perturb it slightly, the robustness is going to take over, the defense mechanisms are going to take over, and it's not going to be a problem. But strong perturbation or persistent perturbation, for instance in smoking, can result in change in the shape of the attractor and rewire the whole network architecture, making it susceptible to future changes. This is just an example of a two-cell network, so you can see what happens in the dark red is high expression of the gene, the sort of white is low expression, and pink is in between. So in the first one, you see high and low expression, it's a stable state. They balance each other out. When you have two mediumly expressed genes, it's unstable, and you can see all the way down. So this is just another way to show you what I mean by these sinkholes. The developmental trajectories, this is a sign of normal development. You see there are epigenetic barriers to the cancer attractor. So in normal development, the system, even if you perturb it a little, is not going to come out. It's got its own attractors here. But if you once change the epigenetics, it's going to land into a cancer attractor. It's going to be very hard to get out. Okay. So, what I'm saying is that cancer involves phase transitions. Transition from healthy systemic attractors to systemic carcinogenic attractors, and here's where the biophysics starts. The healthy attractors, i.e. the body fights cancer, DNA repair mechanisms, apoptosis, programmed cell death, immune mechanisms, tissue repair, tumor suppression, TGF-beta inhibits the cell cycle, and acute inflammation. Carcinogenic attractors, depolarization of epithelial cell potential, which I'm going to talk about, epithelial mesenchymal transition, which is the tumor transition, voltage-gated sodium channel upregulation, cellular and tissue adaptions that promotes tumorigenesis and chronic inflammation. So what I'm saying is that you perturb the healthy systems to a point where there's a bifurcation and you get into cancerous attractors. Okay, so healthy systems have to be perturbed at multiple levels to transition to a carcinogenic attractor. The point of this is targeting one gene or one signaling pathway is probably not the most optimal way to go. And it's kind of like the straw that broke the camel's back. People can smoke heavily for years and not get cancer, but then they get cancer, right? Some of them do and some of them don't. Okay, so where does the biophysics come in? Now we're going to get there. Every animal cell and human cell is enclosed in a plasma membrane which has the structure of a lipid bilayer that gives high electrical resistivity, a low intrinsic permeability. But each cell they have to keep things out that aren't supposed to get in, but they have to let in nutrients and they have to let out the products that they produce for the rest of the body, right? And this happens through ion channels. So every cell has multiple ion channels, and even the mitochondrial membrane has ion channels. And these serve as gates that regulate the flow in and out of the cell. So the term bioelectricity refers to steady-state signals passed between cells and tissues and carried by voltage gradients, ion flows, and concentration domains over a time scale of hours, in contrast to rapid action potentials. So every cell in the body, not just nerve cells, the nerve cells are very fast action potentials, but we're talking about action potentials that hit every cell in the body. So to give you an idea of what I'm talking about, if you look here, amateur radio has a frequency of about 30 kilohertz to 30 megahertz. Radio and TV, 30 to 300 megahertz. The extremely low biological processes that we're talking about are 0 to 300 hertz and have a very long wavelength. So, gap junctions, these are two cells. These are cell 1 and cell 2, and they're connected by gap junctions. Gap junction intercellular communication and connexons are involved in cancer and cancer cell growth control. Spatial gradients of resting potential emerge as systems levels property in multicellular networks. Now you understand why I wanted to talk about systems. So coupling between gap junctions results in multicellular physiological networks with multiple stable attractors in bioelectric space. Are you with me so far? All right. Thus, electric fields are found in and around every living cell and are critical for the proper functioning of biological process. They're not epiphenomena. Changes in electric fields can indicate perturbations in biological functions, such as cell death, where there's a loss of the electric field associated with a mitochondrial membrane, Alzheimer's, a reduction of the electric field associated with a neural membrane potential. So, biophysical microenvironment. The gradients of resting potential are long-range global system for exerting pattern control. As such, they're an important part of the microenvironment, and in many cases, they're upstream from epigenetic changes. They come before the epigenetic changes. Now, this is a double helix. Somebody show me how this arrow works. I don't know how it works. Okay, here's a double helix. And in cancer research, we're always talking about the sequence of base pairs, adenine, thymine, cytosine, and guanine. We don't talk about this backbone here is sugar and phosphate. So phosphate is negatively charged. In order for a gene to express, to mutate, to do anything, genes are wrapped around histone proteins. So there's 8 histone proteins. There's 147 base pairs wrapped around like a thread around a spool. In order for that to express, the DNA has to unwind. It can't be reached by transcription factors or anything else. So, in order for it to unwind, the histone tails on this spool of proteins are positively charged. So in order for the DNA to unwind, the negative-positive attraction between the DNA and the histone things has to be broken. So it's fundamental to the way biology works. So this is a typical epigenetic diagram. You know, once it starts unwinding, the transcription factors come and a whole bunch of changes happen. Now, the key to tumor biology is what we call the epithelial-mesenchymal transition. Most tumors, prostate cancer, breast cancer, lung cancer, colon cancer, stomach cancer, happen in epithelial cell linings. So here, what you have is differentiated epithelial cells which are polarized. And you can read that in every single text on cancer, and nobody seems to be focusing on it at all. They focus on the biochemistry that happens. So what happens in cancer, those cells have to depolarize in order for there to be a tumor. That's what happens. They depolarize, and then there's a whole cascade of biochemical effects which happen so that you get cancer in situ, invasive carcinoma. So you see the depolarized cells, what happens is they de-differentiate. So they go from a differentiated final state to a motile state, where they start humping on top of each other, causing cancer in situ, invasive carcinoma, and then eventually they invade the basement membrane and go out into the system. So what you see, here we go. Okay. So the voltage-gated sodium channels are responsible for metastatic potential. The potassium channels are responsible for proliferation. Sodium-gated channels are not involved in proliferation. The potassium channels are not involved in metastatic potential. However, and this is very important because this is the key to therapeutic implications, any form of depolarization, including movement of chloride, sodium, potassium, or hydrogen ions, and remember, hydrogen bonds are what cause the base pairs in the DNA to stick together. So those are hydrogen bonds. Without any oncogene DNA damage or cancer-causing chemical causes metastatic conversion. This is really important. Any form of depolarization causes metastatic conversion without an oncogene and without DNA damage. This can happen in a normal embryonic stem cell without the epithelial stromal interaction. So this is an issue of the Journal of Clinical and Experimental Oncology that I edited last year. What I want you to focus on is a picture by Mike Levin and Brooke Charnot, who was one of his postdocs. And what you see is in the left-hand side, an oncogene is inserted into the embryo and becomes a tumor, and this is frog larva. Then he put an oncogene into another embryo cell and hyperpolarized it, and there's no tumor, okay? So you look down here and you see, that this is depolarization and this is, this is depolarization, this is hyperpolarization. So what they did to test this is they knew that hyperpolarizing it prevented the tumor. And then they were looking for what is the mechanism. So what they did is they took the hypothesized mechanisms and they systematically blocked them pharmacologically. So they put, in order to hyperpolarize it, they put the embryo in a chloride solution. And so the first thing they did was block the chloride receptors and, the tumor continued to develop. So the mechanisms that were involved were 5-hydroxytryptamine, gap junction channels, voltage-gated sodium channels, and HDAC1. Now, let me see if okay. So HDAC1 is an epigenetic mechanism. Histone deacetylase is involved in all kinds of cell proliferation, cell control. And so if you apply external electric fields, they've been applied to help wound healing, stimulate nerve growth, electroporation of membranes for the delivery of drugs, induce fusion of mitochondria, and stimulate gene expression. So again, I just want to repeat this, that experimentally any form of depolarization causes metastatic conversion, and this can happen without a normal embryonic stem cell, without epithelial stromal interaction or DNA damage or oncogenes. So another thing that Levin did in his lab, he works with salamanders that regenerate. He has shown that regrowth of an appendage in salamander has been induced without knowing or specifying the detailed structure of the organism. So without bioengineering specific subcomponents or targets through the use of bioelectric signal to activate the host coherent self-limiting routine. The reason I started this talk talking about systems and what's not working with targeted therapies is that we're trying to bioengineer subcomponents of a very complex system. And what Levin basically did is he took off the arm of the thing, he caused a tumor to grow, amputated through the tumor, and regeneration normalized the remaining cells. So what he did is he triggered the body's own defense mechanisms with a biophysical signal. So extensive research on the physiology of membrane potential stemming from ion gradients produced by many cell types has led to the knowledge that endogenous oscillatory electric and electromagnetic fields in cells and tissues interact with chemical signaling. So this is the key issue. We're focusing on the biochemical stuff in cancer. It's not wrong, it's just not the whole picture. Furthermore, the physical signaling plays an important role in regulating cell behavior such as differentiation, proliferation, migration, orientation, apoptosis, and gene expression. Now he showed with the HDAC, the histone deacetylase, that was one mechanism for some of these processes. So tumor cells tend to be highly depolarized, whereas terminally differentiated quiescent cells are strongly polarized. So what I showed you in the epithelial-mesenchymal transition, which is key to tumor development of any form of tumor. The relationship is functional, it's not merely an epiphenomenon. Metastatic potential correlates with voltage-gated inward sodium current. Depolarized membrane potential is an epigenetic initiator of widespread metastatic behavior in the absence of a centralized tumor. So it's upstream in the causal pathway. So I'm just showing you that there's a huge number of references for the effect of bioelectric events in proliferation and cell cycle progression, apoptosis, migration and orientation, differentiation and dedifferentiation. Thus, biophysical signals are not just a marker, they have functional significance. However, the cells that overproliferate, change shape, and migrate inappropriately are not necessarily the same cells that are depolarized. And this is what I was saying before. If you initiate a systemic change, what you can see downstream in other cells that weren't hyperpolarized or weren't changed. Experiments have demonstrated that cells that acquired a melanoma-like phenotype were not the ones that had been depolarized. So what you do is you depolarize a system and it has these downstream effects. Only a small number of instructor cells had to be depolarized in order to induce the phenotype in the animal. So not only can cells with the same protein be in different physiological states, and this has not been something that we have understood in cancer, cells in very similar physiological states could reach them by making use of very different ion channels. On longer timescales, networks of voltage-gated ion channels and voltage-regulated gap junctional current pathways can form cell fields. So you have a bioelectric field, which rich feedback dynamics in the distribution of voltage levels that are not at all captured by analysis of protein content. And what we do in cancer research is we analyze gene expression, and now we've gotten more sophisticated looking at systems, so we superimpose proteomics on top of genomics and metabolomics on top of proteomics, but we're still looking at individual expression. So cancer is a reversible physiological state of a multicellular dynamical system. Voltage membrane potential triggers metastatic behaviors at considerable distances. Transplanted or chemically induced tumors can be detected by aberrant voltmeter readings at locations far away from the tumor. And genome-wide methylation changes precede cancer and predict cancer risk. These facts indicate that cancer is systemic even before there's metastases. So, therapeutic implications: cancer is not cell autonomous. Successful treatment requires a systems approach. So prevention through early identification of key biomarkers, obviously that's not new, but these need to include biophysical markers, is the best approach for reversal. Targeting systems rather than single genes or pathways is to be recommended, and we need really urgently a greater understanding of the interaction between biophysical and biochemical signaling in order to improve cancer outcomes. We're looking at the parts of the elephant. We cannot exclude biophysics. So there's also the possibility of electroporation of membranes for the delivery of drugs. So cancer is primarily characterized by global and nonspecific progressive deorganization of the cellular environment, multiple molecular pathways and gene regulatory networks. Tumors can be experimentally both induced and reversed by manipulating signaling interactions, biophysical signaling interactions, for maintaining cell and tissue communication. So, to get to end this, current targeted therapies focus on bioengineering specific subcomponents, i.e. kinase pathways, of very complex signaling processes. But this inevitably has unintended effects on other parts of the system. If we could reboot the system rather than bioengineering subcomponents by perturbing biophysical signaling networks into more a healthy tractor, that may be an alternative. So the last slide, the prospect of using biophysical perturbation to reboot the system towards a healthier attractor holds promising clinical potential, utilizing the body's own defenses to reverse the process of carcinogenesis. So, thank you. That's it. Any questions? Yes?
All these attractors that you can find in the health situation or in carcinogenic situation needs a lot of modeling behind. So you can measure some parameters and then you have somehow to define this situation is an attractor healthy or carcinogenic. Do you know if there are some models, some biophysical models that does this modelization?
So, if I understand you correctly, you're saying there's many subcomponents, and is there a model to identify the subcomponents of this reaction? Did I understand you correctly?
Yeah, more or less yes. But my question is if there are some mathematical models that are able to model these attractors.
So that's an interesting question, and I don't know of mathematical models, but if we look back at the experimental model, this is the experimental model. So if you put the oncogene and you hyperpolarize it, and then you want to see what the mechanism is. So what they did is they took the hypothesized mechanisms, whether it was epigenetic or whether it was gap junction channels or whatever it was, and they specifically, one at a time, blocked each one of these to see if it had a long-term effect. So when they blocked the gap junction channels, they found that they reversed the effect. When they blocked histone deacetylase, same thing. So there was an epigenetic mediator, there was a serotonin 5-hydroxytryptamine mediator, there was a gap junction mediator. So what this shows is that, yes, we can experimentally, and usually when you're modeling something, you model it because you can't do the experiments or you try to project it out. So I think a little more of this work might help with a mathematical model, but I think we need to do a lot more experimental work. Right now I'm talking to Michael Levin and a colleague at West Virginia. We're going to try it in a murine model, a mouse model, because we want to move it into to animal things. Part of the cancer research that I've criticized is transgenic animals. So they modify a single gene and then they see what happens with a drug. Well, we can cure cancer in transgenic animals. A transgenic animal is not the same because you're changing not only a single gene, you're changing the entire system. So the model we're going to use is a wild-type animal that we put in a very well-characterized oncogene in. So I think we need more experimental work is the answer to your question. Any other questions? Okay, thank you.